My name is Banju Singh. I am working as an assistant professor in HR group of institutions, Ghaziabad, in pharmacy department. Today I am going to teach you industrial pharmacy second with subject code 702T. My today's topic is unit 3rd and topic name is Bioequivalence studies. So let's start bioequivalence studies. What is bioequivalence studies? So first of all, I want to tell what is bioequivalence. So bioequivalence is made up of by adding two parts, that is bio plus equivalence, and these two parts have different meanings. It means bio means the term bioavailability. As you know, what is bioavailability? Bioavailability is the rate and extent of drug absorption inside body. And what is equivalence? Equivalence is the comparison of two things. So the both the words by including both words, it became bioequivalence. It means to compare the bioavailability of two different doses or same dose, to compare the bioavailability of two different dosage forms of same salt or two different drugs or different therapeutic activities of two different drugs. So it is a relative term, denotes that a drug substance in two or more identical dosage forms reaches the systematic circulation at the same relative rate and to the same relative extent. As I told, what is bioequivalence? It is that a drug substance in two or more identical dosage forms that reaches to the systematic circulation and at the same relative rate. It means the two identical forms of a dosage form show equal bioavailability inside human body. So the study is known as bioequivalence. For example, if we take there is a drug, antipyretic drug paracetamol. So there are different medicines available in the market of paracetamol. For example, number one is Calpol and next is Dolo and many more. These two drugs are from different companies, but they contain the same antipyretic drug known as paracetamol. So, and there one more drug you can add, crocin. They all three contain paracetamol. So, in bioequivalence study, we can study these three drugs which contain the same drug paracetamol, the bioavailability of crocin, calpol and dolo. We can compare the bioavailability of these three drugs inside humans. So, and what is our aim of comparison of these three drugs is which suits best for humans. It means between among these three drugs which is best for human consumption. So, the main aim of bioequivalence study to uh, exclude the best among various doses forms or various drugs to choose the best. Next, plasma concentration time profiles will be identical without significant statistical differences. It means if we take two different doses of same pharmacological effect or same salt with different doses forms, but they show the plasma concentration time profile identical. Their plasma concentration time profile is identical, but we have to choose the best among these drugs or dosage forms. For in next, so bioequivalence is the study of different drugs or same drug but different doses forms and different drugs with the same therapeutic activity. We have to check the bioequivalence of these drugs or dosage forms, that which is the best for human consumption or use. Next is important pharmacokinetic parameters. Whenever we check the bioavailability inside humans, so in bioavailability we check, let us see, this is a let us see, this is a plasma concentration, this is plasma concentration and this is time. In bioavailability we draw this graph, plasma concentration versus time. Let us make here again, this is time, this is plasma concentration. So, we make a diagram like this. Here it is onset of drug action, this is our C max, this is our T max, this is our area under curve, this is our elimination rate, this is our therapeutic index, this is subtherapeutic, this is termination of action, and this part is duration of action. In bioavailability, we check in this graph at what maximum concentration of drug and at what time it reaches to the systematic circulation in maximum concentration. So, this area is duration of action and this curve is area under curve. Here termination of action, the action of drug is terminated here, and this is therapeutic index of drug. Above this point the therapeutic activity of drug starts. This is onset of action of drug. So, what it said AUC, as I told you, area under the concentration time curve, it is a measure of extent of absorption. It means it shows what? The area under curve, the area under curve shows the extent of absorption. This means this area shows how much drug is absorbed in the body. Next, C max. Here it is maximum. So C max, what I told, maximum concentration of drug at time, at what time the concentration of drug is maximum in body, and what it shows, it is a measure of rate and extent of absorption. It means it is also the extent of absorption in body, but it also shows a rate and extent. Next is T max. At what time the concentration of drug is maximum? So T max is that time. It is the time at which C max is observed. It means maximum concentration of drug is observed at which time? This is called T max. It is a measure of rate of absorption. So, after studying these 3 points, we can conclude what is AUC shows, what is C max shows, and what T max shows. So, AUC is a measure of extent of absorption, absorption of drug inside lumen, and C max is extent of absorption and plus measure of rate, and T max is rate of absorption. It means C max shows both rate and extent of absorption inside lumen. Next, what are the advantages? So, number one advantage, it minimize the effect of inter-subject variability. It means after studying these points, we can minimize the effect of inter-subject variability among different subjects. Next, it minimizes the carryover effect. Third point is it requires less number of subjects to get meaningful results. Next are disadvantages of these studies. Number one, it requires longer time to complete the study. Next, completion of studies depends on number of formulations evaluated in the studies. It means there are number of formulations and we have to evaluate all formulations one by one. So, that will consume our more time. So, the first point here defines it requires longer time to complete the studies because we have so many formulations and we have to evaluate every formulation. Next, it increases in study period leads to high subject dropouts, and because of longer time to complete the study, we have to drop out subjects. Next, medical ethics doesn't allow too many trials on a subject continuously for a longer time. It means in medical ethics generally belongs to clinical trials or non-clinical trials. So, in medical ethics, too many trials on a single subject cannot be allowed. So, we have to take into consideration that these disadvantages should not be done during this study. So, next, what are the requirements for these studies? So, if a new product is intended to be a substitute for an approved medicinal product as a pharmaceutical equivalent or alternative, the equivalence with this product should be shown or justified. It means I have told in bioequivalence studies we do a study of same salt with two different doses, two or more different doses form, or different drugs with same therapeutic activity and different route of administration of same drug. So, if a new drug is intended to be a substitute of a already approved drug, but we want to introduce a new product which shows pharmaceutical equivalence or pharmaceutical alternative. So, with the equivalence with this product should be shown or justified. In next slide, I will describe this point. Next one, in order to ensure clinical performance of such drug products, bioequivalence study should be performed. Obviously, if we are testing two different dosage forms of a same salt, then we have to conclude the bioequivalence studies. If the bioequivalence studies is okay, then only we can send it to FDA for approved to enter into the market. But if it doesn't shows bioequivalence, then we cannot further make any trials or further send it to the FDA, because every dosage forms, every drug first of all should show bioequivalence. Next is equivalence. What is equivalence? As I told earlier, equivalence is comparison between two things or two or more things. So, definition is, it is a relative term that compares drug products with respect to a specific characteristic or function or to a defined set of standards. It means the equivalence or the comparison can be made only a single function or a different pharmacological activity of the same drug. It means we have a already approved drug, but now when we are doing research on the drug, it has shown some different pharmacological actions. So, the new pharmacological action of that drug and the previous actions should be bioequivalence. Next, there are several types of equivalences, several type of comparisons. Number one is chemical equivalence, second is pharmaceutical equivalence, number third is bioequivalence, fourth is therapeutic equivalence. Here, pharmaceutical equivalence means, what is pharmaceutical equivalence? Pharmaceutical means when there is the same salt but two different dosage forms. It means, for example, we in market we have seen diazine tablets and also diazine syrup. Also, there are crocin tablets and also crocin syrup for kids. and like this paracetamol tablets and paracetamol syrups for kids. So, by this formula we can conclude that they are the same salt but two different dosage forms. One is tablet and other is syrup. It means, so we have to show we have to conclude the pharmaceutical equivalence between these two dosage forms. We have to, in bioequivalence studies, we study the bioavailability and other pharmacokinetic factors. The other pharmacokinetic factors are like adsorption, distribution, metabolism and excretion. So, in equivalence studies we study the bioavailability of two different dosage forms and the pharmacokinetic factors, and our aim should be which is the best in two different dosage forms, whether suspension, liquid, ointment. We have to conclude which is the best by studying the bioavailability and pharmacokinetic factors in equivalence studies. So, in pharmaceutical equivalent we study comparison of two different dosage forms. In biochemical equivalence, it means two or more drugs contains the same label chemical substance as an active ingredient. So, we study their bioavailability plus pharmacokinetic factors. Again, pharmaceutical equivalence, I have told same drug but identical in strength, quality, purity, uniformity, disintegration, but different doses forms. Bioequivalence is related term which denotes that the drug substance in two or more identical doses forms reaches the systematic circulation at the same relative rate and to the same relative extent. Next is therapeutic equivalence. It means two or more drug products contains the same therapeutically active ingredient, elicit identical pharmacological effects and can control the disease to the same extent. Here is the example of Covaxin and Covishield. These two vaccines are for COVID-19, but they are two different drugs, but they show the same therapeutic and pharmacological effects. Next type of bioequivalence studies are number one in vitro and number second is in vivo. In vitro studies, we do dissolution studies. Basically, it is done in labs. So, the study is conducted in case of product in solution form or product is having topical route of administration. Product contains no excipients that affect the absorption of API. It means the product has no excipients which can affect the absorption of active pharmaceutical ingredient. Next is in vivo studies, we study pharmacokinetic methods and pharmacodynamic methods. Pharmacokinetic methods, we study plasma level time studies. It is based on the assumption that two dosage forms which exhibit superimposable plasma level time profile resulted in identical therapeutic activity. Two dosage forms, identical therapeutic activity, it means same salt. Next, urinary excretion studies. It is based on principle that urinary excretion of unchanged drug is directly proportional to plasma concentration of drug. Urinary excretion rate is unchanged drug is directly proportional to plasma concentration of drug. Next is pharmacodynamic methods. Pharmacodynamic methods, in this we study acute pharmacological responses of drug and its therapeutic responses, and it is done when? It is done in only in case of local action and no systematic circulation. In equivalence studies, one more study is pharmaceutical alternative. In this, drug is same drug, but it means the salt is same salt, but with different excipients. Excipient is different. This is known as pharmaceutical alternative equivalence. This is also a study of equivalence known as pharmaceutical alternative equivalence. In this, we study same drug but the same salt but having different excipients. It means, for example, we want to make a ointment. So, in first ointment, we add drug plus beeswax, and in the next ointment we add drug plus wool fat. My salt is same, my drug is same, but the excipient is different, and now I have to check the bioavailability plus other pharmacokinetic factors for these two different products. Now they become different products. So we I will study here, the equivalence study will be pharmaceutical alternative equivalence. I will study the equivalence under pharmaceutical alternative. So one more example, one simple example. There is a student and he is studying in maths coaching, in science coaching and in English coaching. So, student is same, means our drug is same, but the excipients are different coachings. So, he can conclude that which coaching is best for studies. So, by doing these by adding these excipients, we can conclude which excipient will be best for our ointment. Thank you.